Good afternoon again, and thank you for joining us for today's EPA Tools and Resources webinar on the detection of the SARS CoV-2 virus in wastewater to inform public health. Today's webinar will focus on EPA ORD's research team's collaborative efforts to reduce uncertainty in wastewater detection of the SARS CoV-2 virus, including method development within our lab, application in sewer sheds with distinctive levels of industrial and stormwater impacts within the Cincinnati Metropolitan Sewer District, and development of wastewater surveillance system in Ohio to support the State Department of Health. If you have a related question during or after the presentation, please type it into the Q&A pod at any time. And once the presentation ends, we will answer as many questions as time allows. We will post the slides from today on the EPA Research website, and Erin will type that address into the Q&A for you. Our presenter today is Dr. Jay Garland. Jay is a senior research scientist in ORD's Center for Environmental Solutions and Emergency Response. He has been leading the EPA research efforts in this area, and I'm just really pleased to have him with us today to wrap up this webinar series for 2020. Thank you so much, Jay, and I'm going to turn it over to you.
Jay, be advised you are muted.
How about now? Can you hear me?
Yep, got you loud and clear.
Okay. And you're back. You can see my you see the presentation mode of the PowerPoint.
Yes, you're all good to go.
All right, great. All right. Well, thanks again. Sorry for the little technical difficulty there. Glad to be with you today. I think Lisa set this up very well, so I don't need to go over the title slide here. We can get on to the content. Some background first on SARS CoV-2 in sewage. It's known that virus is shed in feces by individuals both who are symptomatic and asymptomatic, and that's important, as we'll talk about in this usefulness for monitoring. The level of shedding, though, is highly variable. There's over 4 orders of magnitude based on the relatively limited data that's available, and that's another key uncertainty that we'll talk about. And as background, about 75 to 80% of the U.S. is sewered, and so therefore you can capture and monitor a lot of the population by looking at the sewage. And indeed, early on in the pandemic, the SARS CoV-2 virus was detected in raw sewage, first in early reports in Europe, but then really across the world. And it levels up to 10 to the 7th RNA copies per liter. And when we say that, and we'll get a little bit more into the details, we're talking about detection of the molecular signature for the virus. And it's not detection of infectious virus. And that's the last bullet on the slide, kind of addresses that. There are limited reports of the actual infectious virus in feces, and there's no reports in actual sewage. And it's thought that this virus, the envelope virus of SARS CoV-2, is relatively sensitive to a number of factors, and there's studies actually indicate that it dies actually as it's passing through the colon and under those conditions, as well as when it enters into the sewage itself. So, to be clear, we're not talking about wastewater as a source of contamination and modes of transmission, but instead here, looking at the sewage as a way to inform public health and what's going on in the community. And this is a relatively new idea, but it's not brand new. It's not specific to this pandemic. There were reports about 20 years ago, this idea of using wastewater and surveilling wastewater as a means of informing public health was developed. This researcher, Christian Doughton, was from the EPA, Office of Research Development, and the early focus was on trying to use wastewater to evaluate the rate of drug abuse and use in the community. Subsequent work also continued to look at that, but beyond that, there was also application of looking at the sewage to look for viruses that were spreading in the community, as this paper indicates. And it's also important to note that this approach, this wastewater-based epidemiology, where you're looking at wastewater to look back again at what's going on in the community, it's been used for polio surveillance and actually targeting where there's polio spreading the population and where vaccination programs need to be implemented. That's been going on since the 1980s, and WHO has standard operating procedures on this, as these papers recommend. And then also it's also been used for looking at hepatitis A, norovirus. These are things that actually are found in feces. They're gastrointestinal diseases. And then finally, I show this last slide mainly to indicate that this work was done by one of the researchers in Office of Research Development, Nicole Brinkman, who works with me in Cincinnati. And really, when Lisa said I lead this effort, I talk a lot about it, but Nicole really is the brains and the hands behind it. Nicole and her staff have been in the lab every day pretty much since probably since about late May, working on this. So really, credit goes to Nicole. So why look at wastewater for SARS CoV-2? I think number one, one message is, this doesn't replace all the other things you want to do. It complements other surveillance systems. The advantage of wastewater is that, as I mentioned earlier, it captures asymptomatic and symptomatic people, and it doesn't depend. It's unbiased in the sense that you don't require someone to be tested to show up in the ranks. All it requires is that they contribute to the wastewater system, which most people do, unless they're on a septic system or somehow a separate system. But in a typical city, most people are contributing, and you'll capture that. The idea is that it can be inexpensive, where you can survey a large number of people simultaneously, and then based on that measure, you can then go in and target maybe subsequent individual-based testing. So it can be scalable and inexpensive because of that. And this idea of it being a leading indicator of change in community level infection, we'll talk a little bit about that with some of the data, but there certainly is evidence of that. So just to reiterate this concept a little bit, to frame the discussion today, this quote from Jertan Medema, who's a researcher from the Netherlands who did some of the early work on this, the idea that, again, the sewer is a mirror of society. Look in the sewer to understand what's going on in the society, whether it's drug use or infection rates. And this idea then is this graphically say, okay, how, if you're using it as a mirror, I think the predominant question we have here is how distorted is that mirror, right? Are you looking at a funhouse mirror? And how clear is that reflection of what's going on in the community? And I really feel like what our research has been over the last 6 or 7 months is, you notice these knobs on the side of the mirror, and that they're shown to kind of represent different levels of uncertainty or distortion in that mirror. And we're really trying to dial in and limit that uncertainty and clarify that image of what's in the sewer versus the image back at the community. And we've been working on this, and I hope you appreciate that we are still working on this. Typically in a research world, you would look at all these things independently, and then you would do validation, and then you would start talking about the data to the public. And we're still in the middle of this, and it's been that way since the very beginning. So here we are at the console, dialing in things simultaneously, and we're gonna, I guess I'm inviting you in a little bit today to kind of see how we're working these dials as we go along. So for the presentation, these different topics I'm going to cover in the first 3 bullets are really correspond to those knobs or those uncertainties. You need to understand the variability or uncertainty in the analytical method itself. We're going to talk about that. But even if you understand that, you need to understand what the dilution and degradation of the signal is by the time you measure it in the sewer, right? So if you want to go back to what was kind of donated or put into the sewer, you need to understand what happened between the point of discharge and the point of collection. And then further, even if you understand both the analytical method development and that the dynamics in the sewer, you also need to understand, try to relate that signal back to community infection rate. And I think the take-home message I would tell you all is that, this is one imperfect measure of community infection, and we relate it back to other imperfect measures of community infection, right? Because the data we see about infection rates, those are all people who have been tested, likely people who are symptomatic. Sometimes they're asymptomatic, but it's not the true number of infected people. So there's all those layers of uncertainty that we need to kind of resolve. And the other point I'd like to make for those 3 bullets is, if you're really going to address this issue, you need to cut across those 3 different discipline areas: environmental analytics, sewage and sewage engineering, right? And then finally, public health officials. So this work that I'm going to talk about really does cut across the analytical laboratories, the utilities, and the public health officials, and that is critical to really making progress on this. And because of that, I want to emphasize and kind of close with talking about the state network that we were able to work with, or how do you pull together a network, how do you bring together those different elements, and then also talk about how to translate this information into public health decisions. Because regardless of how confident you are in the signal, how clear your reflection is, it's still the true value of it is if it's going to be used in public health decision making. So I'm going to talk about those different elements. So first, let's start with analytical methods. I know this slide has a lot on it, and it just kind of illustrates the different elements of when we say the analysis. It involves sample preparation, typically sample concentration, because we want to concentrate the sample to improve detection of the viral signal. Then once you've concentrated the sample, you have to extract that RNA, and then finally you have to detect that RNA. So depending on your level of experience with these, some of this is going to seem very basic. I just try to introduce this overall concept, though, for those who may not be as familiar with molecular analysis, environmental molecular analysis. So some of the issues, so from the sample type, all of the samples I'm going to talk about, it's untreated wastewater. We really want to get it prior to treatment, right? Because you really want to look at the signal, as clear a signal as possible. You could look at primary sludge. It's untreated, but what we've dealt with is just the composite sample that utilities typically collect of their untreated waste. And when I say composite samples, these are taken over a 24-hour period, and then they take these samples anyway for a variety of permitting purposes and looking at the contaminants in wastewater. So it's a very easy way to interact and collaborate with utilities, because we're really not asking them to do more than they already are doing. Now, that said, there are a lot of applications where you're trying to sample not at the treatment plant, but more upstream in the sewer network. And in that case, you may not have a composite sample. You may do a grab sample. You may do some compositing, but not over the 24 hours. So there's some of the issues there. In terms of the processing, how you handle the sample, we did some early work on that. Refrigeration is important. You can typically, if you refrigerate it, you don't lose signal over the course of 2 or 3 days. So taking samples, keeping it refrigerated, and shipping is possible. Another question there is the matrix spike, and we'll talk a little bit about that. Adding in some kind of organism to look at your recovery efficiency of the methodology. When you concentrate the sample, there's a number of different ways to do that. I'm going to talk about those in a little bit more detail later on. So kind of pass over that if you're not familiar with PEG precipitation or ultrafiltration. The idea is you're trying to get viral particles that can be passed through normal filters because they're small in size. So you need to have ways to concentrate that out of your sample. And in terms of extraction, there are different extraction protocols for removing and purifying the RNA molecule, because this is a RNA virus. And then in terms of measurement, there's different platforms for doing this. I mean, their commonality is that they're amplifying that signal. So you have a certain sequence for SARS CoV-2. Specifically, I'm going to talk about the N1 and N2 genes, which are the N1 and N2 genes related to the spike protein, that little red knob that you see on the pictures of SARS CoV-2. So you've got a specific sequence for the genes that create that spike protein. And you can measure that. You want to amplify it. So polymerase chain reaction allows you to amplify that signal, but you want to do it in a quantitative way. And there's quantitative PCR, qPCR. There's also digital droplet PCR, which is what we use, it's just different. Without getting into the details of those methodologies, they're just different machines and platforms to do quantitative measurement of the amplified signal. And then finally, some other considerations. I'm going to talk a little bit about the need for QA, QC, and also this idea of the biosafety. So let's talk about that first. So first of all, you're dealing with wastewater. So anybody dealing with wastewater, when I say the wastewater.
Sorry to interrupt. Can you just minimize that black Teams box in the corner?
Sure thing.
Thank you.
Yep. So in terms of biosafety, when I say the wastewater risk is the same, it means that there is all wastewater is considered a biosafety level 2 media and sample, and you need to handle it with a biosafety level 2 procedures. Typically, involves a number of things, but given the fact that this is a respiratory virus, and even though I've said that we don't have clear evidence that it is the pathway for transmission and that there isn't live infectious virus in the wastewater, to be safe, and the CDC recommends that you use typical biosafety level 2 laboratory procedures with additional or biosafety level 3 precautions, and that really is respiratory protection, as you can see in this figure or this picture of the person doing the extraction. And it's important to do this as you're doing those initial stages of centrifugation, membrane filtration, because you can create aerosols. So you really want to minimize inhalation risk there. So that's kind of what we do, and we're using a biocontainment suite in our laboratory here in EPA to do that. So this is our overall processing scheme. And as I mentioned earlier, we mainly start with 24-hour composite samples from the wastewater treatment plant. The first thing we do is we will spike it. I mentioned the matrix spike. So the OC43 is a spike. OC43 is a human coronavirus. It causes a common cold, so it's relatively safe to work with. So we spike that sample in, spike a known amount of that in, then do a direct extraction of the sample without any processing, and that allows us to get a percent recovery based on how much we added in without any of these further steps. Then we will look at centrifuging the sample and getting out the large solids and looking at, are there particles, virus associated with the large particles, then doing the membrane filtration and looking at viral particles associated with smaller solids, and then the remaining liquid, which could still contain viral particles because of their small size, do ultrafiltration. And then we extract and analyze each one of those fractions, do the RT digital reverse transcriptase, which means we're looking for the RNA, and we do digital droplet PCR in that. And then we look at this range of targets for that sample, including, I mentioned the N1 and the N2 gene, as well as the OC43 to look at our percent recovery, as well as other fecal indicators. And I'm going to talk a little bit about that a little bit later, about how do you estimate what the fecal strength of your wastewater is. And that's important because you can get dilution again in the wastewater due to inputs that say rainfall, depending on what your system is. And we're going to talk more about that. So overall method performance metrics, our limits of detection are about 655 RNA molecules per liter. So when we see a detect, that's kind of our minimum detectable level. The recovery efficiency, which I mentioned, for the matrix spike, the beta coronavirus OC43, we get about up to 50% recovery. And I'll mention a little bit later some of the variations in approaches that have led to different levels of percent recovery of that beta of that OC43. The endogenous viruses are those fecal indicators that I mentioned. Crass phage is a phage. Phage is a virus, but it doesn't infect humans. It infects a bacteria that is thought to be specific to humans. So one of our intestinal flora, a Bacteroides species of bacteria, this virus infects the bacteria. So you can look at that presence of that virus as an estimate of how much fecal load is in that sample. That one we get a very good recovery out of. The other organism there, the PMMOV, that's the pepper mild mottled virus, which is, interestingly, this is a pepper virus. It infects peppers, so it's not a human virus. It's not endemic to humans because of any association with our own microbiome, like the crab's phage. But it does show up in feces a lot when you sample relatively large populations because of the level, apparently, of peppers that we eat, and you can see that in the waste. So we look at that as well. The other issue about QA/QC is not just the recovery efficiency, which is your overall measure of recovery, but you also there can be different steps in the process that will limit your efficiency. One of those is inhibition of the digital droplet PCR itself. So when you do that, because it's enzyme driven, there can be certain compounds in the wastewater that would inhibit it. The good news is what we've seen to date is very minimal inhibition of PCR. So again, sorry if this is too far in the weeds on methods for some, but for others, I think what I'm trying to emphasize here is the QA/QC and knowing these limits, very important, regardless of what specific approach you may be using. Trying to understand these factors, both the recovery efficiency, inhibition, limits of detection, that's what's critical for any method that is being applied. I talked about supply chain, so let's talk a little bit about that briefly. A couple examples of supply chain problems. One is the ultrafiltration step. We were looking at all these fractions originally, but now we've eliminated the ultrafilter fraction because the ultrafiltration unit that we use, these millipore centrifugal units, they're these basically small units that allow us to do this. They became became out of supply. They were unavailable due to supply chain constraints. Fortunately, this graph here shows the proportion of the RNA that we recovered in our samples based on those different fractions I mentioned, the pellet, the filter, and the ultrafilter fraction. The centricon is to capture that last bit, what's remaining after you've taken the pellet and filter. Fortunately, as you can see, the bottom line here is that 90% of the measurable virus is in the pellet and filter fraction. So thankfully, that supply chain disruption was not a problem. In fact, it streamlined our analysis. We still recover 90%. The second disruption that we had is that the extraction kits that we use for each one of those fractions, they have become limited. There's a lot of people doing this, and there's certainly a limitation. And we ran out of those back in late October, so we switched our extraction process to using triazole chloroform extraction, and then following with RNA precipitation. So we've kind of moved away from this kit that was available. This is kind of the old school way. Though people who did molecular biology before the kits, this is kind of the approach they used to look at RNA extraction. And what we found, fortunately here, is that when we moved to using the triazole, we did some initial comparison of recovery efficiency with the QIAGEN kits versus the triazole, and what we found was about a 10-fold improvement in recovery efficiency. So in that previous slide where I said our recovery efficiency was about 50%, that's up to 50% with the triazole method. So in this case, our response to the supply chain issue constraint actually led us to a better path to a certain extent. So we're now using the triazole. So again, in the weeds of methodology, but important elements of understanding the clarity of your signal. But now let's move to the issue of sewer dynamics. So let's get out of the analytical lab and into the sewers, and this is just as important. And in fact, I know more about the sewers of Cincinnati now because we work closely with the Metropolitan Sewer District of Cincinnati because they are true collaborators on this, because they know their system. And what this graph shows is that within the basic city of Cincinnati, which is outlines, pretty much the city of Cincinnati overlays with Hamilton County pretty much exclusively. This is actually the outline of Hamilton County, but it's also covers the Metropolitan Sewer District of Cincinnati. The red dots are the main treatment plants, and the two treatment plants that I'm showing here, or that are shown, that Mill Creek is the largest, and at the bottom there it kind of compares the descriptions of some of the characteristics of those. Mill Creek is 118 million gallons per day. It represents about over 400,000 people contributing, so it's very large. It also has industrial input. It also has a large amount of combined flow, meaning that there's storm sewers. So it can be diluted, and it's variably diluted based on the amount of stormwater runoff. So it's useful to look at because it's a large footprint for the city. But on the other hand, Taylor Creek, which is just to the left there, this sewer shed smaller, but also no industrial flow, no combined flow, and because of that, it's not diluted at all. And the dilution reported there, that's the kind of the overall strength of the sewage based on BOD. That's the value for that. Typical wastewater for that sewer shed based on an average value. So you can see Taylor Creek is actually, it's concentrated and so it's not diluted. So we are really working with all of these sewer sheds to a certain degree, but predominantly Mill Creek and Taylor Creek I want to talk about because it allows us to look at this fecal normalization issue to a certain extent. In addition to that, I also mentioned that you may want to look inside the sewers, or inside the sewer shed. So here I've outlined Lick Run, which is one community within one neighborhood within Cincinnati, and it's called a sub-sewer shed. And we sampled this because early on in the pandemic, we wanted to identify potential hotspots in the sewer network. At that point, back in late May, we were having a difficult time picking up any signal. As you'll see from later the slides, that's not a problem now because of the increased prevalence of the virus in the population. But because of that, we were thinking about, were there sentinel sites where you might see higher levels of the virus? And we picked Lick Run because it's a high density community, also a lower socioeconomic condition. So back in late May, these folks were more frontline workers. They did not have the luxury of working from home, and therefore they may be more exposed to the virus. And then when they came home, they were living in higher density housing and therefore more risk of spread. So we wanted to understand that. So in this case, when we looked at Lick Run, this is looking inside the sewer shed, not at the treatment plant. In this case, for Lick Run, it was relatively unique because there's a large combined sewer overflow within draining that or servicing that area. So we were able to tap into Lick Run at that point. That's what that large structure is on the left-hand side. Typically, when it's not activated, there's a flow in the bottom that's relatively low. When rainfall overwhelms the flow, then it opens up those gates and you get a combined sewer overflow into the Lick Run waterway there. So this just shows you, in this case, instead of doing a 24-hour composite sample, we do about a 3-hour composite sample in the morning to try to capture not the full 24-hour contributions, but the high flow period in the morning. So the next slide here, let's talk about some data. And this is data looking at, what you're looking here is the lines on this graph. The red and green lines are of the N1 and N2 genes that I mentioned earlier. So that's our estimate of what the level of SARS CoV-2 is in the wastewater. The bars are the 7-day average of case rates within those specific sewer sheds. So you can see it's different from Mill Creek and Taylor Creek. And this is an important point. We now, through our collaboration with Ohio that I'm going to talk about next, we can get the case rates by sewer shed. So when you're trying to understand the relationship between the sewer levels and estimating how that relates to the infection rate, you really want to compare apples to apples where you can, not looking at the level in Mill Creek versus the entire county, which would be all of the people within the county, not just the people in Mill Creek. This data is for infections within those. And so what you can see here is that this is data from, again, early May through, this data only goes through about middle of October. I'm going to show you some data that's even more current than that. What you can see here is that we had a peak in a peak in cases in early June, and indeed we did see rising levels of SARS-CoV-2 in the wastewater. Same thing with Taylor Creek. The level wasn't as high early on, but you notice also that there was this secondary peak, which unfortunately continues to grow. And you also can see the relationship between the levels in Taylor Creek and both Mill Creek and Taylor Creek, and the relationship between the sewer levels and the case rates in that sewer shed. The data on the bottom kind of illustrates again, you can directly correlate those values, but you also want to account for potential variation in flow, because if you're getting dilution of your sample due to some factor like rainfall, that's going to give you going back to the mirror analogy, that's going to distort your image and not reflective of what's going on in the community, but it's just the rainfall event has nothing to do with infection rate. So what we're doing now is looking at, if you correlate N1 to the infection rate, for Mill Creek, we get about a 0.43 correlation. It's significant, but you notice within Taylor Creek, it's a higher correlation because, again, going back to the previous slide, Taylor Creek is a system that has no combined flow, no industrial flow, so it should show a clearer relationship without any normalization for flow, and that when you try to normalize, when you do normalize, for Taylor Creek, it really doesn't improve the relationship, but on the other hand, it does improve the relationship to a certain extent by normalizing that N1 to, say, crass phage, which is that, again, that fecal strength. So we're, again, going back to the analogy of we're in the middle of this and working these knobs, we're really in the middle of writing all these results up. We're looking at the correlation straight out between N1 and infection rates, but then also normalizing to the flow volume that's collected at the treatment plant, as well as the normalization to the different estimates of fecal strength, crass phage, PMOV, in addition, HF183, which is a bacterial specific fecal indicator. So we're in the process of doing this, and this is very important to try to normalize, and this is important for any wastewater-based epi work, this idea of, since you're taking this composite measure, really having a good way of normalizing for your population size and uncertainty in your population size. Trying to use fecal indicators is a good way to do that. Now, let's talk about this idea of, and looking at a little bit more data here, let's talk about this idea of sentinel sites a little bit. The data I'm showing here, and this is only from early May through mid-October, so the same time period. And what I'm showing you here are the red lines in each one of these graphs is what the peak was, the peak infection in the overall Hamilton County, the entire district there that I showed. So all those major treatment plants, all of the sewer sheds in Hamilton County. And what this shows is that if you look at Mill Creek, which again was the predominant one, up over 400,000 people in that sewer shed, the rise in levels, again, RNA molecules per liter in the wastewater seemed to really reflect pretty spot on when that peak was in the overall community. When we looked at Taylor Creek, which was a little bit lower, it lagged behind that. But you saw the data that I showed you that in that case, Taylor Creek, the rise in infection was a little bit delayed in that sub-community. But then Lick Run, again, that low-income, high-density housing, when we began to look at that, our first samples were from early June, it was already higher than any other sample we'd already measured, indicating that there was higher infection levels in that community. So, and in fact, we knew that because working with the local public health agency, they had seen higher levels there. So what this shows you is that, if you want to look for sentinel sites, let's say you don't have, early on in the pandemic, when you don't have all that data, you don't know what the distribution is, and you want to try to say, okay, what's my best place to sample? Well, you could say the best place may be the largest one to capture the most people. Mill Creek, captured that. I think it, was a good relationship. If you wanted the idea of a sentinel and really early warning, this idea of trying to pick a more at-risk community of high density, low income might be valuable. But you notice in this later larger wave that's happening now across the county, you see Taylor Creek, which did not show that early warning, an early outbreak in early May, it rose and it's at high levels even before, and rose even before Lick Run did. So what that shows is that the factors driving the differential spread of virus in the community are going to change over the course of the pandemic. Early on in May, late May, early June, when we saw this rise, well, Lick Run probably was more at risk because they weren't not working from home, but the personal decisions that people are making now that are leading to spread of the virus are different later on in the pandemic than early. So one last data slide, not to overwhelm you with data. This shows you all the data that we are collecting in the ORD labs across Cincinnati, which is Mill Creek, Muddy Creek, Taylor Creek, Little Miami, Those are all of the main sewer sheds that we're monitoring. And then we're also, in collaboration with the state of Ohio for their network, which I'm going to describe in a second, we're looking at Dayton and western and eastern regional plants around the Dayton area, as well as the cities of Springfield, Marion, Portsmouth, and Hamilton. These are all smaller cities in southwestern Ohio. Take-home message from that is that you can see, and we talked about some of these early rises, like the peaks that there did seem to be correlation, you could use it. The wastewater did reflect relatively minor increases in infection rates, but what you can see is that across the board, in all of the cities, there are much higher levels now. Ohio, unfortunately, is at a much higher level of community spread now than it was at any time before, and in fact, certainly the wastewater reflects that. So the take-home message is that there was a range of minor peaks early, and you could see those in the wastewater, and now there's just really strong, consistent increase across communities. So what does this data mean? Well, a little bit, just kind of, well, then we're going to talk about the translation to the state, but if you want to relate the SARS CoV-2 wastewater data to actually the number of infected people, I was talking about correlations. It certainly correlates with the rate of infection. But if you actually want to say how many, especially how many people are infected, I'm going to take this number in the wastewater. You need to know your measured concentration. You need to look at the recovery efficiency. So if it was 50%, the real number is twice what I measured. You also need to understand what the dilution occurred, what the decay is. Maybe due to just temperature dependent decay or industrial discharges, etc. You need to know all those things, and even if you know all those, to get at the accurate number of people infected, you need to know how many SARS-CoV virus are shed per gram feces, and that, as I mentioned in the early slide, is very uncertain. So that first bullet, I think this is a useful goal, and there are people developing those models. But the point right now, the focus is really saying, let's look at relative changes as a way of monitoring what's going on in the community. And so that's what the Ohio Wastewater Monitoring Network is really doing. And just a little bit of background, this timeline on that. It was started, the governor in Ohio, Governor DeWine, really at the end of May expressed an interest in implementing this. Ohio was able to use CARES funding to do this. The funding went through Ohio EPA, but Ohio Department of Health is really the project lead. And then they also have used the Ohio Water Resources Center, which is Ohio WRC there at the bottom. And this is an entity at the Ohio State University that its mission is to integrate across the state university. So this was important because we needed to set up a network of laboratories to really do the sampling and the intensity of sampling that we wanted to do. So beginning in July, we targeted the 7 large cities in Ohio, and that was with OSU, Ohio State University, University of Toledo, and the US EPA. We kind of contributed those southwestern parts of the state, and those other labs did the central and northern parts of the state. And then with time, that's grown. This graph is even outdated. I talked to Ohio Department of Health yesterday. It's now up to 62 sites. So here we've added, in addition to large cities, medium and smaller cities, and also additional laboratories. So we have a number of labs now that are working. So one of the important things in this laboratory network is once a month, we send the same sample, hopefully from some positive site. We go on a monthly basis and say, what's the highest level in the state, and take that sample and split it and send to all the labs so that we can compare results. And what you can see is there's about, this graph, what it shows is that for the 4 labs that had been doing analysis up to this point, there was about a log difference between labs. Some of the early labs that kind of come on, they're a little bit further out of that range, 2 or 3 log units out, and we really work with them to try to dial that in. So I would say on average, we see about a log unit difference between the labs in the split samples as they come on fully on board. And so that's important to understand if you're trying to compare across cities. You're trying to compare Cleveland to Cincinnati, for example. But it's important that the state, they are, again, looking at the trends, and the same lab always does the same site. So you're really then just looking at the interlaboratory variation. While important to understand and evaluate the new labs as they come on, the method they're using, it's also important to kind of use the same lab at the same site. So this network, again, they're focused on the trends or significant changes. The the kind of rules and criteria they're using now is that we sample twice a week. So if we see 3 successive days that are at least a 10-fold log increase, that really, the ODH then works and notifies the local health district. They provide information to them about how to interpret the data. They also have a messaging toolkit, which I'll show on the next slide, which actually kind of allows the local authorities, how to use this data and how to message it. And then they also notify the pandemic testing team and to establish pop-up testing to increase testing at that location. So in talking with ODH, Rebecca Fugit, who's the lead from ODH on this, they have contacted over the course of the network since its implementation, they've notified local health departments 100 times now. And in terms of that, the request for pop-up testing, they've implemented that 40 times. So the figures here, these spots just represent all of the sites that are being monitored in Ohio. And then if you look at some of the, especially the larger cities, you can see when you zoom in, you can see the footprint of the sewer shed. So the blue outline is actually the actual representation of the sewer shed that is being monitored. And like I mentioned, now Ohio also has the infection rate, the number of people infected by sewer shed, allowing us to do those direct relationships of apples to apples, infections in the sewer shed to the wastewater levels. And then if you're interested in any of this website, the address there at the bottom. So in terms of these public health applications, I mentioned that the toolkit, there's the link to that toolkit if you're interested in looking at it. Ohio also, in addition to what we're talking about now, they're also doing more focused monitoring of multiple sites on campuses to support the college and universities as they come back online in January. And this idea, they're going to do up to 5 locations on campus, really kind of looking at dormitory level monitoring. The idea here is screen level assessment of the wastewater. If you see a positive response, then kind of go in and test everyone in the dorm to really then isolate who may be asymptomatic and pull them out and quarantine them. Also should note that we work with CDC quite a bit on this. We've been interacting with them from the early days of the pandemic. Ohio is one of the early adopting states, and they're coordinating with CDC on reporting the data as into the National Wastewater Surveillance System, which CDC is developing. And there's the address for that as well. And this is a unified, kind of consistent way to monitor, to report the data. And then it's also housed at CDC in addition to at the Ohio level. And that's something CDC is working with. Ohio is the first state to report, or first state to report to that. Wisconsin and Utah are soon to follow. And I think there are other states, obviously, that would implement it as well. So speaking of collaborations, we here in ORD, I mean, that's kind of a lot of what we're about. And we try to collaborate not only with our federal partners like CDC, as I mentioned, as well as the Ohio network, which I think I've described, right? We both supply samples, or we analyze samples for the state network. We've also provided information on sample processing, like the refrigerated hold time study we did early on to share with the group. We kind of facilitated the early interlaboratory comparisons. So that's a big part of the collaboration, and very, very great collaborators. I mean, it's like, I can't say enough about the Ohio Water Resources Center, Zuzana Boarova, who's leading it for them, and Rebecca Fugit. They've really been the clear leads on the Ohio effort, but we've been collaborating with them. The public utilities I mentioned, they're key to this as well. All right, so Cincinnati MSD, they're a true collaborator on this because they provide that wastewater engineering. But in addition to that, we work across the state with the Ohio utility organizations. We reached out to them early in the pandemic to kind of, even before we were sampling any of them, just kind of get them primed for this as a possibility and educate them on what this was and what it wasn't. It wasn't the infectious virus monitoring. And then we've also worked with different states as they brought on Arkansas, Maryland, New Jersey, New Mexico have all reached out to us relatively recently as they develop plans. And all of those states, they, instead of the community level work that I talked about today in Ohio at the sewer shed level, all of those states are focused more on congregate housing settings. Either, maybe it's nursing homes, but also maybe it's a lot of work going on in correctional institutes, as well as the universities that I mentioned. And then in addition, we are, in contact with the research community. Water Research Foundation had an initial summit on this we participated in. They had an interlaboratory method development study, which we contributed to. I mean, we participated in, and then we've also kind of reached out internationally as well. So let me just summarize, kind of try to tie all this back up. Going back to those 5 points that I mentioned, in terms of analytical method development, I'd say, there's no standard method. I mean, like I mentioned with the 6 labs in Ohio, they're using different methods. And having many options available may be useful, particularly to address the supply chain issue. If everybody's using the same methods with the same supply requirements, then you really run a greater risk of kind of being dead in the water at certain times. So I think there's some value in that, given the nature of this pandemic. But while they may be different methods, they have consistent quality control for assessing method performance. As I mentioned, recovery, efficiency, inhibition control, as well as the detection limits, et cetera. So really understanding your method, use different methods, but understand the QA/QC. This dilution and degradation in the sewer, like I said, we're ongoing comparison. I think this idea of how to best normalize the data is important, especially as you move into the sewers. If you're doing monitoring away from the treatment plants, you don't have the luxury of having their volume estimates for that 24-hour composite. You need other ways to look at fecal strength. So the correlations that I mentioned with PMOV and crash-phage, we are really working hard on trying to get that data finally finalized and published in the very near future. And now, in terms of degradation, like I said, there are existing temperature-dependent decay rates for coronavirus that could be used. We also we in Cincinnati, we're doing some targeted studies on industrial waste and what's the impact of those. And then, as I described earlier, you can look at all those factors, dial all those variables in terms of relating the sewer set to infection rates. You really then additionally need additional rate on shedding rates. And I would add here, though, that like with Ohio, and I think on the CDC News website too, this idea of being able to get infection data by sewer shed and doing a lot of relationship between the sewer signal and the infection rate there, that's going to do that's really going to inform this relationship quite a bit as well. Then finally, in terms of developing the network, I can't emphasize enough, I think it started that way. I mean, linking the wastewater utilities, the environmental analytical labs, and public health agencies, that's what this is about. I mean, you got to do that. I would recommend, if you're at all thinking about this, really reach out to the other individuals to pull it together. And then the network of labs are important to increase capacity if needed, but build in QA/QC to understand the comparisons. And then in terms of translating the data to public health, as I mentioned, focusing on the trends or significant changes to reinforce public messaging, that's important. As these these models to predict infection are refined, I think those may be also useful with this general trend analysis to look for increasing levels and reinforcing messages, even though we all know that there's a good deal of pandemic fatigue right now. And in terms of early warning, like I mentioned, I would say when we say early warning, and you looked at some of the data I showed, it looked like there was relatively reasonable correlation between the level of wastewater and the level of infection in that particular sewer shed. Maybe it could be a little bit leading indicator, but the key variable is how quickly you're getting any of those data back. Because what I was showing you was archive data in a way, right? After the fact, after we finalize the infection rate data, the wastewater data, the real key is what data do you have quickly? So the turnaround time is really pivotal and something we get pressed on a lot within the Ohio network. Ideally, we would like to be able to get the data back within twice weekly sampling within 24, 36 hours would be great. And that's really pushing it. And then the idea of sentinel sites may be very useful, but try to understand the attributes of those sites. What may be driving spread of the virus early on in the pandemic versus later. As an example, you really need to understand those factors. And then finally, this idea of right now targeted sampling to direct individual testing and actions, that seems to be a very clear path of relating this data to decisions. Not just reinforce public messaging, but really saying, okay, I've got this composite sample. I know somebody's positive in this dorm or in this area, group of people. Now I need to go in and test everybody. That's very important to optimize the use of of this wastewater signal and our testing resources. And boy, you know, looking back, I think about if we could have the data we have now back in April and May from the wastewater, that's when it would have been particularly useful, right? I mean, thinking back then where testing was very limited, long-term rounds. If we had this kind of aggregate sampling of wastewater, that would have been so valuable to both understand where the virus was spreading and to then also target testing and really most effectively utilize those resources. So I think one of the goals, kind of thinking forward to not if but when the next pandemic happens, and I don't mean to sound negative, but I do think there's a strong possibility that this isn't the last time. Question of how can we be best prepared so it's not taking months to roll this out, but we could get this kind of wastewater-based surveillance in place very early on. So please feel free to reach out to me with any questions. And again, shout out to all the people that have been working on this, both within our